Welcome back. In this last chapter, chapter 43, we're going to talk about the applied in environmental microbiology, and we are going to discuss about three sections. The 43.1 and 43.2 will discuss about the water treatment, that the potable water treatment and the wastewater treatment. And in the section 43.4, we are going to talk about some of the biodegradation, bioremediation, which is also very important for our environment. So the first section, we are going to talk about that how we purify and the sanitary analysis of the ensure safe drinking water. And this is a huge issue in the whole world because more than 1 billion people actually do not get safe drinking water. And this is an essential commodity for the human life. So that is very important. So we are going to discuss about this. So the first thing is that the water purification do have some microbial contaminant candidate, and this list is listed by the Environmental Protection Agency in the United States. And this gives us the idea that what are the potential pathogens that can survive in the water and may pose a health risk. And due to that reason, we have to purify the water. And this is also very important or critical in controlling the waterborne disease. And as we can see that in different countries, particularly the developing countries and the third world countries, these water purification system is not adequate enough. And that's the reason that there is a lot of waterborne infection and diseases that all the time happens in these countries. On the other hand, usually most of the developed countries do have a robust water purification system along with regular monitoring. And this actually goes into the municipal level, that how your water department take care of this water that is supplied to your house all the time. So let's take a look at some of the microbial water contaminants which are listed by the environmental protection agencies. And again, this is very self-explanatory, but just giving an overview, these adenoviruses that cause respiratory illness, sometimes some of these cold-like symptoms, common colds, all the time, it is not caused by rhinovirus. Adenovirus can cause that as well. It can also give you some kind of what we call as a stomach bug, gastrointestinal disease. Calciviruses, also called self-limiting nausea, vomiting, diarrhea. Norovirus is one of them. And then I'm just skipping those. Enterovirus, which is a group of virus which gives you enteric disease. This includes the poliovirus, Coxsackie virus, and echovirus. And I will just explain that what it is here. On the other hand, there are some other which is not mentioned here, but it is relevant in other countries, is called rotavirus. So this rotavirus is also another enterovirus which causes gastrointestinal disorder. Now, we are all vaccinated against the polio, so this is not a cause, but polio actually goes through our gastrointestinal tract and cross the intestinal lining, and then finally it makes its way into the nervous system. But this is not of a concern for us anymore because we all are vaccinated against the polio and it's almost considered as eradicated as such. But one of the important things that I'm just going to point out, because you already have that virology chapter, when you look at all these viruses, which are adenoviruses, calciviruses, and all these enteroviruses, all of these viruses are naked or non-enveloped virus. And why it is so? So just have an idea that if you have an enveloped virus, it do contain a membrane which is usually stolen from the host cell as the virus buds out of the cell. And this membrane is made up of a phospholipid, which is very labile to any kind of harsh condition, which could be just simple soap water. And on the other hand, think about that all these viruses need to go through your stomach, which do contain pH 1 or 2 due to the hydrochloric acid. And all viruses which are enveloped viruses will be easily destroyed in your stomach. Whereas all these naked viruses do have this capsid made up of these proteins, and these proteins are sometimes extremely stable even in harsh conditions. So it is much more difficult to destroy or inactivate these viruses, and that's why they can cross the stomach, which is full of hydrochloric acid, very low pH, and still can cause infection. Just as an idea that we are just nowadays talking about the coronavirus as well. Coronavirus is an enveloped virus. That's one of the reasons that if you take precaution, just simple soap water or just hand sanitizer can easily inactivate that virus, but not for these naked viruses, which doesn't have the envelope outside them, just only have the protein capsid, which is much more robust compared to all other enveloped viruses. Then you have many different bacteria like Campylobacter jejuni causes nausea, vomiting, and diarrhea. And then you have an E. coli. This is the enterohemorrhagic E. coli, which causes the bloody diarrhea. Sometimes this causes kidney failure and it could be life-threatening. This is the O157H7, means oligosaccharide 157 and hemagglutinin H7, this specific strain. You also heard about Helicobacter pylori. This is a bacteria which basically lodges into the mucus layer of the stomach. So this Helicobacter pylori do contain a glycocalyx outside them, which is rich in the polysaccharide, and that prevents the hydrochloric acid to access the bacteria so it can survive in the stomach environment in that harsh condition. And often these are implicated in gastric ulcer as well as cancer. Then there is hepatitis A virus, cause liver disease leading to jaundice. So this is also another virus which can cause leading to jaundice as well. Legionella pneumophilia is a bacteria that can be found in the hot water system, causing bacterial pneumonia when inhaled, usually limited to elderly patients though. Then you have some Mycobacterium avium that can also cause respiratory illness. Then you have this Naegleria fowleri protozoan found in warm surface groundwater, cause primary amoebic meningoencephalitis, which could be very, very dangerous as well. So this is a protozoa. Along with that, you have something like Salmonella enterica cause GI illness. Severity depends upon the serover. Say, for example, serover typhi of the Salmonella enterica, that will cause the typhoid fever. Very, very severe. Shigella sonnei, this causes the GI illness, including the body diarrhea. So these are some of the microbial water candidates which are listed by the EPA. There are many other as well, but they are not of a concern in the United States. They are mostly found in some other countries and they are not tolerated at all. So there is also some list. Say for an example, Giardia. So Giardia intestinalis is a protozoa which can cause gastrointestinal disorder. Entamoeba histolytica is a protozoa which can also cause very severe type of gastrointestinal disorder, but these are not very common in the United States. So here we are looking at the different steps of the water purification. It's very self-explanatory that how they are being treated, and we'll again go into a little bit more detail. So you have an untreated water, and these untreated water is held in a container. This is basically for the color and the precipitate removal. And here you can just undergo aeration, chemical oxidation, ion exchange, sedimentation in this process. From here, the water is then moved into the second container where you have this softening of the water, where we remove this calcium as well as a magnesium. And there is a chemical precipitation happens at this stage, and dosing, mixing, flocculation. And sometimes there are particularly some material like alum, which is aluminum hydroxide, is added, which is a flocculation agent, and that can precipitate many of these chemicals. So after that, what happens that you have the turbidity removal, where the chemical coagulation also helps in the flocculation, settling, and filtration in this process. And then after that, you have an aeration and chemical oxidation and absorption, where you remove the taste and the odor. And then finally, you have a disinfection stage. Either you have irradiation or ozonation and chlorination. This last disinfection stage will remove the rest of the microorganisms. So what are the water purification steps? So the first thing, the water is held in high levels of suspended material in the sedimentation basin. And you can see that right here, the large particles settle out. And then this is a repetition that partially clarified water in that mixed with alum and lime in the settling basin. And more of the material precipitate out by this coagulation and the flocculation process. This removes the microbes, organic matter, toxic contaminants, and suspended fine particle as well. And then the water is then flowed through these rapid sand filters, which physically trap the fine particle and the flocks, and then water treatment with disinfectant causes chlorine and ozonation that we just talked about. So what we saw in the last picture, this is basically the clarification of that. Although most of the bacteria removed in this process, but it cannot remove all the protozoa. Say, for example, it cannot remove the Giardia cyst or Cryptosporidium oocysts or Cyclospora or many of the viruses. And these are some of the concern, because what happens that the water which comes into the primary clearing agents is usually many times is from a surface reservoir, say from a lake. And Canada geese are actually a problem which do bring many of these protozoal contaminants into those surface waters, which can pass through our water purification steps. So these are some of the concern that we do have. So next, what happens that the EPA has developed a regulation which is called a long-term 2 enhanced surface water treatment rule. And this process sets the maximum containment level goal for the specific pathogen. Say, for an example, MCLG, this is called MCLG, say for Giardia, Cryptosporidium, or say Legionella and certain viruses, this is 0. That means that there is any trace amount of Giardia, Cryptosporidium, Legionella will consider that water is not potable at all. There is no safe level if there is any of these that I said, the Giardia cyst, Cryptosporidium oocytes, Legionella, or certain viruses are present. This is also 0 for total coliform, which is an indication of fecal contamination. What do we mean by that? That a fecal coliform is basically a bacteria which is present in our gastrointestinal tract in colon, and these are usually rods, and coliform means their natural habitat is in a colon. Now, if those bacteria are present and if we identify, usually that tells us that water is contaminated by some fecal contamination, and that is not tolerated. It has to be tested for all other possible fecal contaminants as well. So these are some of the safety goals within the EPA regulation and the guidelines. So health goal is set at a level which no known or anticipated adverse effect on health of persons occur, which allows an adequate margin of safety. So after that, we are looking at the different purification method. The precise treatments depends on the level of contamination, but generally involves filtration techniques. So one of them is a slow sand filters. Water passed over the bed of sand with a microbial layer, biofilm, cover the surface of each sand grain, and the waterborne microbes are removed by adhesion to this biofilm. So this is very effective. And then further inactivation of the viruses can be achieved by chemical oxidant, high pH, photooxidation, and we have already seen that again, chlorination, ozonation, these are also procedures to remove these viruses. So how we can check the condition of the water, or this is the sanitary analysis of water, so that all the municipalities constantly do this sanitary analysis of water, and there is always a microbiologist present in these departments to do this routinely. And this sanitary analysis of water is based on what we call as an indicator organism. So these indicator organisms that we said, this one of them is the coliforms. Coliforms indicate the fecal contamination of the water supplies, and this indicate possible contamination by human pathogen as well. So what is an ideal indicator organism? It should be suitable for analysis of all types of water, present whenever enteric pathogens are present, survives longer than hardiest enteric pathogens. These are gastrointestinal pathogens. Does not reproduce in contaminated water. They are harmless to the humans, and its level in water reflects the degree of fecal pollution or contamination. This is very, very important. And there should be a assay procedure for indicators should be great specificity, and the testing method should be easy to perform as well. So these basically is a rule how we look for the contamination in the water, and we'll just explain that in a second. So the two commonly used indicators which do have all these ideal indicator organism property is the coliforms. When such foreign enteric indicator bacteria are not detectable in a specific volume, and this is generally 100 ml of the water is considered potable or drinkable. This include the bacteria which are not limited to intestinal tract, so tests have been developed that focus on fecal coliforms, which are the fecal contaminants. There is also another is a fecal enterococci. This is increasingly used to test the brackish and the marine water. Just giving you an example. Usually, sometimes when you go to the beach, usually after a heavy rainfall, sometimes you may see that there is a flag in the beach which says the water is not safe for swimming. And what does that mean? That means that particular municipality actually checked that marine water, and due to the rain, excess amount of these fecal enterococci drained from the land into that brackish or the marine water, and it does contain unusually high amount of these particular enterococci or a group of organism, and due to that it is not safe for the swimmers to do that, and usually they will get dispersed and the bacterial level will come down to a particular safe level. And this happens all the time, we have seen, and usually this is very common just after a sudden rainfall. This is a typical thing that where this increased amount of fecal enterococci actually do increase in the brackish and the marine water. So what are the different types of tests that we perform? So we'll spend some time on this test, that how we do use these indicator performance and how we actually do this test in the laboratory. So for an example, as we said, that this is a multiple tube fermentation technique and used for many years for sanitary analysis of water, and it's a kind of a golden standard. So first you take a water sample, that will be the 100 ml. And then what you can see that these are the different broths. Either they use this lactose broth or they use this lauryl tryptose broth. Lauryl tryptose broth is used particularly along with the lactose, which specifically isolate or allow the growth of the fecal contaminants or fecal coliforms. That's what it is used. Now you can see that there are many different types of dilutions. This is a double strength broth. The reason is a double strength because in these you are using a large amount of inoculum, like a 5 or 10 ml mixed with 10 ml of the sample. Whereas here you are using the single strength broth because you are just inoculating 1 ml here. You're just using 0.1 ml here, so it will not dilute the broth too much. But here, as you're using 10 ml, you just use the double strength of the broth. Now, after inoculating these, what you are seeing that each one of them inside the tube or the test tube do have a small glass tube. This glass tube is an inverted glass tube. Think about that. This is a mini micro test tube inside a test tube. So this is called, there is a particular name. This is called a Durham tube, like the Durham, North Carolina. This is called a Durham tube. And this Durham tube is used for trapping gas. So what will happen that if the coliform, all of them in presence of this lactose and this lauryl tryptose broth will produce gas. And if they do so, this gas will be trapped inside this inverted Durham tube. That's the reason that you have that tube. So now what you do that you incubate them in 35 degree, 37 degrees Celsius for 24 hours. And then if you do not see that there is any gas in here, then you know that there is no indication of any gas formation, so probably it is not contaminated, or there is no coliform present in there. If you see that there is a bubble, which is indicated by just this small clear area, if there is a small bubble inside that Durham tube, that means that there is some gas formation by the bacteria, and that is a positive indication. So at that time, what is done, that this particular bacteria is, or this broth, is inoculated into something which we call as a brilliant green bile broth. So why we call this brilliant green bile broth? Because you have bile, salt in there, which promote the enteric bacteria or the fecal coliform to grow. And also the brilliant green actually suppress the growth of any gram-positive organism. So it's a selective broth. And also it contains the tube. So if there is no color change in this broth and there is no gas, then you can easily see that right here you have a negative result. On the other hand, if you see that after incubation, this broth color changed to yellowish color, and also right here you have gas bubble, then you know that there is a fecal coliform growing. So at that time, what you do that you take this organism and you plate it into any of the two types of plate. And these plates are two types. One could be EMB, which you call it as a eosin methylene blue agar plate. So the eosin methylene blue agar plate, which contain the lactose in the plate as well as a sugar, and that's a differential and selective media. So what the EMB does, EMB will only allow the gram-negative bacteria to grow. So it will not allow the gram-positive to grow. So it is selective for the coliforms or the gram-negatives, which are only gram-negatives. There is no gram-positive. And it will also give you a colored reaction. So all the coliforms, which can basically break down lactose, will produce a purple color. On the other hand, particularly the E. coli, if present, will produce a greenish metallic sheen. So you almost know that there is some bacteria which are gram-negative fecal coliform bacteria is growing in there. On the other hand, there is another media which we call as a less endo. This is developed by a company called Hardy Diagnostic. And why you call it a less? Because it is a formula which was used or initially developed in the Lawrence Experimental Station. So alias endo or Lawrence Experimental Station formula endo. And that endo agar is also another selective agar as well as differential agar. And that produce a reddish colony with a metallic sheen, which is an indicator of a gram-negative lactose fermenting bacteria. So after you isolate that, these are all first round of confirmatory tests, first round, second round, third round. Then what you do that you inoculate that again back to a lactose broth to see that whether it produces a gas or not. This is the same experiment. You are again repeating this experiment. And then also you inoculate this bacteria into a slant. And from that slant, you allow it to grow for 24 hours. You take the bacteria, make a slide, and do a gram staining. And in the gram staining, if you find that there is a rod-shaped organism which is non-spore forming, you know that you have a fecal contamination of coliforms in your sample. So this is the multiple tube fermentation test routinely done. Now, this is pretty time consuming. It is routinely done, but it will take several days. You can see that. So there are some other methods as well. We're going to look at them as well. So the other test that is done is a membrane filtration technique, presence or absence PA test. We are going to talk about that. And this ColiAlert test, it is from coli or coli alert, from their ColiAlert defined substrate test. And also there are some molecular analysis like a flow cytometry, FISH, quantity PCR or microarrays. But the molecular analysis is mainly for the research purposes or epidemiological purposes. These are not routinely done at the municipal level as such. So what are the things that can be easily identified though? Say for example, Legionella pneumophilia, that can be identified by quantification by the PCR techniques. On the other hand, the 16S ribosomal RNAs are very specific to the coliforms. They have very specific sequence. Those primers can be used for differentiating the non-pathogenic or enterooxygenic strains of the Shiga toxins. E. coli, say for an example, there are all different E. colis, but there are some specific E. colis which do contain this Shiga toxin like the O157H7. You do have some specific primers because this E. coli contains some specific proteins, and you can identify the presence of that particular E. coli in your sample by this specific PCR technique, which is kind of not possible for here. So think about that your normal E. coli, which is not the bloody diarrhea causing E. coli, will come out as a positive test. On the other hand, O15787 will also come out as a positive test, but you cannot distinguish in between those two E. coli. But here, the PCR technique will be able to distinguish in between a pathogenic strain and a non-pathogenic strain. There are some other tests as well. Like, say, for example, this is routinely done. This is called a membrane filtration technique. So these membranes are 0.2 micron filters or bacteriological filters, which we talked about somewhere else. And these are made up of nitrocellulose, polyvinyl pyridine, many different types of material. And water is passed through these filters. And once you pass this water through these filters, and this is usually under a vacuum, the bacteria will be trapped on top of the membrane filter because the pore sizes are 0.2 microns. So the bacteria will not be able to pass through them. Now you take the filter and you place it on the growth media, incubate, and see that whether there is any colony or not. And then from those colonies, you can detect them total coliforms, fecal coliforms, or fecal enterococci by all these different types of tests that we discussed. So that's your membrane filtration technique. On the other hand, there are some presence or absent tests, and this is a modification of the most probable number. So this uses a larger water sample, about 100 ml, and the sample added to a lactose-containing medium. Remember that all the coliforms use lactose, and also do contain a pH indicator to detect acid production. And based on the assumption that no indicator organism should be present in 100 ml of drinking water, this test is done. So how this test is done? So this test uses a broth, which is a lauryl tryptose broth. And again, this lauryl tryptose broth particularly select the coliforms. Also in this broth, there is an indicator, which is bromocresol purple. So this bromocresol purple will turn into an yellowish color if there is any fecal contamination. So the positive test, so this bromocresol purple is purple in color, but a positive test or presence test will produce an yellow color, and that will again tell that there is presence of some fecal contaminant or the coliform, and then you can do some confirmatory tests. There is another test, and this is commercially available. This is known as the Colilart defined substrate test. So here you have also a 100 ml of sample, and this is added to a medium which actually do contain either this ONPG and the MUG. And we are going to talk about that. What are these? So what you're seeing that this is a typical sample bottle, and this is basically simple test used to detect coliform fecal contaminants, about 100 milliliter water sample. That's important. So this is right here. These are the typical bottles. In the sample bottle, the first one is a sterile or uninoculated sample. This is a control. On the other hand, the second bottle contains a media that uses ONPG, which stands for orthonitrophenyl beta-D galactopyranoside. And what it is, that it is an artificial substrate for a coliform enzyme, which is galactosidase or beta-galactosidase. So all the coliform, including the E. coli, will have this beta-galactosidase. So if the E. coli is present, the ONPG is going to be used as a substrate, and the E. coli enzyme, the beta-galactosidase, will convert the ONPG to orthonitrophenol. And this orthonitrophenol does have this yellow color. So presence of this yellow color indicates that there is a coliform contamination in that water. There is also another test, and this uses another enzyme with a different substrate. So this substrate is MUG, which is basically a methyl umbelliform beta-D glucuronide. So this MUG, or this particular chemical, uses a particular enzyme that is also present in these coliforms, which is beta-glucuronidase. So if there is any fecal contamination, and if the bacteria which are producing beta-glucuronidase is present, they are going to convert this MUG into a fluorescent compound. And again, it will just not fluoresce. You have to place this bottle over a source of ultraviolet. So in the presence of an ultraviolet, if you see that there is a blue fluorescence, that indicates that there is a fecal contamination or a coliform contamination, particularly E. coli. So the next part that we are going to talk about is the wastewater treatment, which is maintains the human and environmental health. So that is very important. So all these wastewater, this is from basically our flushing the toilets, from our sink, from our daily usage that we use, from our shower. All these different wastewaters needed to be treated properly before it goes or released into the environment. And we are going to talk about that how this is done. And we are going to see that this process of the wastewater treatment do have a primary, secondary, and a tertiary wastewater treatment. Primary and secondary is absolutely essential, and the tertiary is usually a little bit more expensive. And in some areas, depending upon the ecosystem, it may or may not be used. And we're going to talk about that. So there are a number of steps that are spatially segregated, and this decreases the organic matter and the number of microorganisms in human waste impacted water. And this lead to a major reduction in the spread of the pathogen in the environment as well. So these are the common contaminants of the water. And this is, again, I'm not reading each and every line. This is very self-explanatory. So you can easily see that what are the suspended solids. So you can easily see that there are biodegradable organic compounds, protein, fat, carbohydrate. Think about that. These are from the sink. These are from basically the toilets. Then you have the pathogenic organism, which can come from human waste as well. The priority pollutants are produced. These are organic, inorganic compounds, and these are industrial products. And many of these could be toxic, carcinogenic, mutagenic, and teratogenic. And along with them, there are refractory organic compounds. This could be phenols, surfactants, agricultural pesticide. Think about that. When you talk about the phenol, just your home Lysol, this will be considered as a refractory compound. Surfactants, that's detergent soaps, these are considered as refractory organic compounds. So from the liquid that is going out of your washer, it contains those. Then you have these agricultural pesticide. These are all toxic compounds from the environment and also resist the conventional wastewater treatment. So we are going to look at these, that how these could be detoxified in the 43.4 section, bioremediation, that chapter. Also, you have the heavy metals usually discharged by the industry, discharged into river streams is toxic metals. So these are toxic to all trophic levels from the primary autotrophs to the secondary and tertiary heterotrophs as well. And also you have these dissolved inorganic constituents, calcium, sodium, sulfate may be added to domestic water supply and may have been removed prior to the wastewater usage as well. So these are some of the contaminants of the wastewater, and we're going to see that what we can do to treat these wastewater treatment. So the first we are going to talk about the primary treatment, which removes the solid material and forms the sludge. And the secondary treatment is dissolved organic matter is transformed into what is called the microbial biomass, and also carbon dioxide is liberated. And it forms a stable flock, settle well, and the bulking sludge, and this doesn't settle properly though. And we are also going to talk about this activated sludge system in the secondary treatment. So here, the important part is that the microorganisms play a critical role in functioning in the activated sludge system. So what you are seeing that right here is this wastewater effluent in this beaker on the far left contains bacteria, other organic material that resist the settling. In this case, a commercial coagulant like a ferric chloride is added. Second beaker right here. And when you add that, it accelerates the formation of stable flocks that is settled at the bottom of the beaker. And then on the far right shows the effluent once this settled material is removed. So this just gives you an idea that how the wastewater is actually treated. So let's take a look at the industrial scale, that what happens, that this is a typical sewage treatment plant. So just giving an overview of that, and we are going to talk about that initially when you have all these sewer systems bring the material to these plants, usually there are some coarse filters. And these filters out like 14, 15 millimeter fine mesh, large plastics, debris, these are filtered out so that there is no clog that happens downstream. Then there is a filter which can vary depending upon 6 millimeter to even 0.035 millimeter. And these 6 millimeter to 0.035 millimeter is also a secondary filter which filters this material. From there, the material goes into what is called a primary clarifier, and we call this as a grit chamber. And sand, bone ash, these are used to reduce and settle down the material. And from those primary clarifier, then it goes into the secondary aeration chambers, and this secondary aeration remove the hydrogen sulfide usually, and also reduces the biological oxygen demand. And we're going to talk about that is the biological oxygen demand. And finally, that goes into the secondary settling tanks that you can see that these open settling tanks, these are the large open ones. And then from there, chlorine is added, and then these are released into the environment. The sludge can undergo anaerobic digestion in these chambers and also basically thickens. And finally, you dry the sludge, and the dry sludge is usually hauled away, which are used mainly for the burial or disposed into the landfills and can be used as a fertilizer or soil conditioner as well. So let's take a look a little bit in detail. So what we are looking at, the trickling filter is a waste effluent passed over rocks or solid material with microbial biofilm. The microbial community actually degrade the organic waste by this process. And this is an aerobic process of the secondary sewage treatment. Also, sometimes an extended aeration, which is shown here, and this also reduces the amount of sludge produced by using microbial biomass for energy requirements. So what we are looking at in these different things. So right here, we are looking at these activated sludge with microbial biomass recycling right here is maintained in a suspended state to maximize this oxygen, nutrient, and waste transfer process. So this is the effluent that goes out. And then from here, it can go into a trickling filter. So this is the trickling filter after this, where the wastewater flows over the biofilms attached to the rocks and other solid support, resulting in the transformation of dissolved organic matter to new biofilm, biomass, and carbon dioxide. So the excess biomass are then treated into the final clarifier. So right here, this shows an extended aeration process where the aeration is continued beyond the point of the microbial growth. And this allows the microbial biomass to self-consume due to microbial maintenance energy requirements. And this extended length of this reactor allows and this process of biomass self-consumption. And in this process, sometimes the minerals are originally incorporated in the microbial biomass and are released to the water as the process occurs. Next, what we are looking at, that the anaerobic digestion, often sludge from the aerobic sewage treatment together with the material settled out in the primary treatment, are further treated by anaerobic digestion. And this reduces the amount of sludge for disposal and also produce methane. So this is basically a basic design of an anaerobic digester, which includes an inlet for delivery of the waste. And in the first step, what happens that there is a fermentation of these complex substrates happen, and it produces a lot of different types of organic acid like butyrate, propionate. So remember that these are like butyric acid, propionic acid or propionate, lactic acid or lactate, succinic acid or succinate, ethanol, also acetic acid. And along with that, of course, carbon dioxide and hydrogen will be produced. And these are all the products of primary fermentation, which is the very first step. And these are done by the anaerobic Clostridium, bacteroids, Streptococcus, then Peptococcus, Eubacterium, Lactobacillus. All these are responsible for the initial fermentation. Once this fermentation happens, there is a second step, which we call it as an acetogenic reaction. In the acetogenic reaction, which are mainly carried out by the Centrophomonas and Centrophobacter, what they do that they consume these other organic acids like propionic acid, butyric acid, succinic acid, and convert all of them to acetic acid or acetate and hydrogen and carbon dioxide. And then finally, in the third step, the archaea comes in. So in the archaea, they convert these hydrogen as well as the carbon dioxide into the methane. And there are a number of these archaea, and all of their name basically start with methano prefix, like Methanospirillum, Methanococcus, like those. And then finally, once this is done, the effluent goes out, and this takes almost 3 to 4 weeks. This whole process it's a long-term process. And also in this process, the methane is utilized or it is captured to this methane outlet. So this is the anaerobic digestion to reduce the sludge amount. So the tertiary treatments, what happens, that this is the removal of the nitrogen and the phosphorus that may promote eutrophication. So eutrophication is basically what it is, that when large amount of this nitrogen or phosphorus is released into the water bodies, you can see that these water bodies will promote the algal growth due to this high amount of nutrition, which are detrimental for the nature. So that's the reason that phosphorus is usually precipitated as calcium phosphate or iron phosphate. And the nitrogen, excess nitrogen, is actually removed. Say, for an example, there is ammonia that is being produced, that is a nitrogen source, and the ammonia is broken down in high pH, and the nitrogen gas can be released from there. So these are some of the processes which can be carried out by some of the microbes as well. And then at this step, you can also use some of the carbon or the charcoal filter, which can also remove heavy metals, biodegradable organics, many remaining microbes or the viruses as well before releasing it to the environment. But this step is very, very expensive. And this is only used if there is a reason before the release of the wastewater in the environment requires this specific step. Otherwise, this step is usually omitted because it is not cost effective. So the next step that what we are talking about, before we release the material or the wastewater in the environment, there are some testing that's needed to be done. And this is a testing for the water quality, is the measurement most of the time is how much of the organic matter is being released in the environment. So are we removing the organic matter from the wastewater or not? That is the most important thing. So there are several ways to do that. The first one is called the TOC, or the total organic carbon levels. This is basically done by the process of this oxidation of the carbon to carbon dioxide, and this is measured by an infrared method. So TOC levels is very important. The next one is a chemical oxygen demand, or COD, similar to TOC, but will not measure the lignin, which is basically organic compound, which can be measured here, but this will not measure the lignin levels. But it will measure all other organic material or the carbon from other organics except lignin. The next one is BOD. This is also a very common thing, which is a biological oxygen demand. This measures the carbon oxidized by microbes in 5 days or 20 degrees Celsius. So, as we have said, as the wastewater moves from the primary to the secondary chambers and the process goes on, as we have seen, the biological oxygen demand is also reduced. And that means the organic material, as the wastewater is being treated, the organic material amount is also going down. And that is indicated by the biological oxygen demand. These methods don't measure the nitrate, phosphate, or sulfate levels, which may cause eutrophication, which are the algal growth contributors. But there are several methods to basically reduce these nitrate, phosphate, or the sulfate levels from the wastewater as well by the tertiary treatment that we talked right here. So other than that, there is also a home treatment, and there are many areas, particularly in the remote areas, which doesn't have a public sewage system. The septic tanks are used for sewage. This functions as a simple aerobic digester and sometimes frequently fail to work properly, contributing to groundwater contamination. So you can see that the wastewater from the house So here you can see that this system combines an anaerobic waste liquefaction unit, this is the septic tank, and with an aerobic leach field. So basically in here, the oxidation of the liquefied waste takes place in the leach field unless the soil becomes flooded. So a well-drained soil is necessary for the proper functioning of the septic tank. Otherwise, it may fail. So one of the biggest challenge that providing clean drinking water and wastewater treatment for 7 billion people in the world is a challenge. So there are some water reuse, purification of wastewater into the drinking water. This is only used in California, not in other states so far. And this is a complex process, and this need to be required to remove the pathogens and all pharmaceutical traces from the potable water. Also, the sewage overflow during the storms due to the stress capacity of the municipal system is a growing problem. And this construction of overflow basins, increased infrastructure is often the current solution. And this can happen during a hurricane or a tornado or a storm surge, that the sewer overflow can contaminate the local area. And many times these are some of the reason that can bring some of these contaminants to our supermarkets. Because think about that a sewage overflow in a field which is growing some of the greens like your lettuce or other kind of spinach, those kind of things. And if after that sewage overflow, the greens are harvested or the produce are harvested from those contaminated fields and brought to the supermarket, there's a chance of contamination. These are some of the challenges that is there. So the 43.4, in this section, we are going to talk about what is the biodegradation and bioremediation. And this is the microbes' power to clean the environment. And this is one of the process that is utilized, and we're going to talk about this. So the biodegradation and bioremediation, these are the metabolic activities of the microbes. This can be exploited in natural environments. And some of the examples, the microbial communities to carry out biodegradation, bioremediation, environmental maintenance process, and we're going to explain each of them. And addition of the microbes to soil or plants to improve the crop production. So these are some of the processes that we can utilize, and we'll discuss about these in this chapter. So the first thing that what we are looking at is a biodegradation. And why we need to use this word or use this process, the biodegradation is very important because there are many different types of chemicals. When they are released into the environment, they are toxic. They can pose serious type of environmental toxicity and pose threat to the environment. So we need to degrade them. And in this process, microbes do help. And there are some minor changes, fragmentation, mineralization. So what happens is that right here in this process, we call this as a dehalogenation. And you can see that right here, this chlorine is being removed from this. So this is replaced by an hydroxyl group. So this is basically a dehalogenation. Remember that chlorine, bromine, and fluorine, these are the three halogens. So chlorine is one of them. So this is dehalogenation. So in the functional group, which is attached and substituted by the hydroxyl group for the chlorine group. And the B is a fragmentation. This is actually a breaking of the organic compound into organic fragments in such a way that the original molecule could be reconstructed. So this is another process, and this may be less toxic. And the next one is the mineralization, is the complete degradation of an organic compound into the inorganic minerals. This is the most complete of all the three processes. So let's take a look at the bioremediation. So the bioremediation is the removal of the toxic industrial products in the soil or aquatic environment. And it is, of course, a daunting task, but it is necessary. So there are many industrial compounds like perchloroethylene, which is PCE, trichloroethylene, TCE, polychlorinated biphenyls, which are called PCBs. These are some of the common contaminants, and removal of these contaminants are necessary from the soil and the aquatic environment. And the degradation of the complex compounds requires several discrete stages, and these are usually performed by different microbes. So the initial contaminants are converted to less toxic and they are more readily then finally degraded. So the first step for many contaminants include, say, for example, organochloride pesticide, alkyl solvents found in the paint solvents, aryl halides, is a reductive dehalogenation. So what is a reductive dehalogenation? This is the removal of the halogen like bromine, chlorine, or fluorine, while at the same time adding electrons to the molecule. This can occur in two ways, either by hydrogenolysis. This is seen in this reaction right here. And this reaction shows that 1,2-dichlorobenzene is dehalogenated by alkyl hydrogenolysis. Alternatively, there is also another process which we called as a dihaloelimination. In this reaction, you can see that there are two adjacent carbons with halogen, and in this process, the two chlorines are removed from them and a double bond is introduced in between these two carbons. So this process do require an electron donor, which usually mostly water. And there are some specific type of genera of bacteria can do this. These are usually carried out by some major genera like Desulfobacterium, Desulfospirillum, Desulfomonile, and these are really implicated in this process. So the reductive dehalogenation process that we have seen can occur in anoxic condition, and this anoxic condition is just the soil humic acid. So the humic acid is an ideal environment for this, and the humic acid can act as an terminal electron acceptor as well. So the anaerobic dechlorination of the vinyl chloride and dichloroethane is degraded by this anoxic reductive dehalogenation. And after this anoxic step, the main structure of many of the pesticide or xenobiotic is often more rapidly degraded in the presence of oxygen or in the aerobic condition. The reductive dehalogenation occurs in anoxic conditions, and often the soil humic acid do offer the ideal environment for this and acts as a terminal electron acceptor. And for example, anaerobic dechlorination of vinyl chloride and dichloroethylene is degraded by this anoxic reductive dehalogenation process. And after this anoxic step, the main structure of many of the pesticide or the xenobiotics or the foreign material often proceeds rapidly in presence of oxygen as well. So what is the fate of a chemical in the nature? So the structure or the stereochemistry actually do play a critical role in predicting the fate of a specific chemical. So the meta effect occurs when the constituent is in meta as opposed to the ortho position, and the compound will be degraded at a slower rate. We're going to take a look at this. Say, for an example, there is a common lawn herbicide which is known as the 2,4-D or 2,4-dichlorophenoxyacetic acid. The chlorine is in 2,4, which is the ortho position. This can be degraded, or the 2,4-D can be degraded in just one summer. But on the other hand, what happens, there's another product which is known as 2,4,5 trichloro, 3 chlorines, trichlorophenoxyacetic acid, where you have 1 chlorine, 5, that is in the meta position, not ortho. 2 and 4 is in ortho, 5 is in meta position. So what happens, that can persist in the soil and will not degrade so much. So what happens that there are some positive and negatives for these. So this 2,4,5 trichlorophenoxyacetic acid is used to control brush control, and it can remain in the soil for years. On the other hand, 2,4-D can be degraded in one summer. But what happens that if you constantly use this 2,4-D, there could be a change in the ecology of the microorganisms in the area where 2,4-D is routinely used. And these microorganisms can degrade the 2,4-D even quicker. But what can happen that suppose these are supposed to control weeds for 3 months or 6 months, instead of that, you may see that the weeds are appearing within a month or two. And these are due to the change in the ecology by using the same kind of herbicide year after year. So many compounds that are added to the environments are chiral. That means they possess a symmetry and handedness. They have a right and the left-handedness. And microbes often can only degrade one isomer. So these are called the D or L forms. This is, you have seen that in amino acids as well. So say for example, for the human beings, we can utilize the L form of the amino acid, but we cannot utilize the D form, whereas many microbes can utilize the D form as well. So depending upon this same kind of thing, the chirality or these isomers, some of the isomers are effectively degraded by some of the microbes, but some of them can't. Unfortunately, almost 25% of the herbicides are chiral compound. So a particular isotype which is biodegradable by certain microbes are more friendly to the environment. But also another thing, what happens, as I said, the same thing with what happens with the ortho and meta. If you're using a particular compound in the environment, the ecology in that area changes where the microbes can degrade that chemical in the nature much faster, losing its effectiveness. So you have to find a fine balance for that. So on the other hand, what we can see that the downside of biodegradation can lead to a widespread damage and financial loss if it occurs in inappropriate situations or uncontrolled manner. Examples are metal, especially iron pipes, toxic degradation products. These are some of the examples. So now some of these examples that we have seen. So this is a compound which is called the DDT. This was routinely used as a pesticide. It's no longer used, but this could be degraded into a compound which is called the DDE, which is equally toxic. So what happens is that this is an interesting story that you can read a book. It is written by Rachel Carson, and this was published in 1962. And the name of the book is The Silent Spring. And she was a marine biologist, but she just excellent writer. And this book basically tells the horror story of this DDT and the environmental suffering from the DDT. And this was the first movement that started in the United States to ban this compound, and it was completely banned. And this particular book, which is The Silent Spring by Rachel Carson, is in 2012 designated as a National Historical Chemical Landmark by the American Chemical Society. So it was a huge impact on the environment, and we no longer use this. So here is another example of trichloroethylene or TCE. Unfortunately, this is also widely used in different solvent, and this is degraded into the vinyl chloride, and this is also a very toxic carcinogen. So these are some of the downside of the biodegradation. Even after biodegradation, the product can be toxic. That is the take-home message from here. So bioremediation also involves the stimulating degradative activities of the microbes already present at the contaminated sites. So what happens that sometimes you can just add fertilizer, nitrogen or phosphorus to supply them and help them with this co-metabolism technique. So addition of easily metabolized organic matter, just like a glucose, increases the degradation of the recalcitrant compound, not usually used as a carbon or energy source. So that's basically the overall process of the bioremediation. What you are seeing in this picture is that this is a picture which is due to an oil spill in 1989, a ship which is owned by the Exxon, an oil tanker, Exxon Valdez, that ran aground into the Prince William Sound in Alaska. And it spilled more than 10 million gallons of oil, which actually affected about 1,300-mile shoreline and heavily affected about 200 miles of shoreline. And in Alaska, it caused a huge amount of environmental damage from seals to different types of marine animals. And what you are seeing right here, that they are basically spraying these nitrogen phosphorus based fertilizer to speed up this biodegradation process. And there are rich hydrocarbon degrading microbes in there. And by limiting these nutrients, this bioremediation process can be sped up. So here is a typical way that how the oil degradation basically happens. So here you can see that the primary oil degradation involves a bacteria that starts the biodegradation process. And these are in the blue microbes. These are the ones which are the oil digesting microbes. And these bacteria release a variety of degradation products, and they are farther broken down by secondary consumers, and indirect interaction influence also the rate of bioremediation. So initially, this mineral weathering or phosphorus-rich chemicals can be utilized in this degradation process as well. And once that process do start, the primary oil degradation will produce this degradation intermediate, which can be further broken down by the secondary consumer. Unfortunately, what happens that these primary or the secondary consumer can be attacked by this protozoan predation, or these can be lysed by these phage that can reduce the amount of oil degradation as well. So these are some of the drawbacks of the process. The bioaugmentation is, on the other hand, say a microorganism to a complex microbial communities, and this is usually for a short-term increase in the desired biodegradative property. And these outcomes provide the protect microhabitats. Say, for an example, a surface of a seed, root, or leaf, inert microhabitats, say, for example, microporous glass. These are some of the examples. So what happens, just to give you an example, sometimes just the septic tanks can be added with a culture, and that will enhance the silage formation to improve the efficiency of the septic tank. Sometimes if there is a small oil spill or something, a hydrocarbon spill, sometimes in this process, some non-pathogenic bacillus, wetting agent, nutrients can be used to degrade this or disperse those oil at a small scale. This is called the bioaugmentation, usually in a much smaller scale to speed up a process of biodegradation. This is actually a very beneficial process. Many microbes do produce acids, and they can solubilize the desired metal for recovery. These bacteria are used in mining areas with levels too low for recovery by smelting, or from the ores which are not much rich. So these are carried out by, say, Leptospirillum species or Thiobacilli to recover copper. So basically what they do, that they release these acids and they convert the copper ore into copper sulfate, which can be recovered, and from copper sulfate, the copper can be extracted. So these bacteria can recover almost 70% copper from the very low-grade ores as well, and these are also used in the mining process as well. So this is the overall idea about this, that whether it's the water we drink, wastewater, or contaminated environments, there are many needs for cleaning up the different areas or the items to have a proper ecological balance. And this is basically the take-home message. So this will be the end of chapter 43.